What is going on, folks? I don't know if you can hear me. You can maybe let me know if you can. From my end, it looks like everything is good. I'm actually going to move this. Sweet. I am being heard. Move this chat over here so I can see it. Sweet. You can hear me. All right, sweet. Let's go through this. We're going to just kind of work through some of the different stuff. My goal here is to just kind of work through the different units. And as you guys have questions, I can answer them. And it's funny. I have looked back at some of the live streamers, like the dude who does the history stuff, and saw his chat, and you couldn't even read it. It was so fast. And so thankfully mine's not doing that. That's good. All right, so we're just going to work through these units. Actually, you know what? I'm going to go back to Unit Z here because we can talk about a few things. I think we've talked quite a bit about graphing, and so I don't want to go over too much of this, other than to say that make sure that you label your graph and that you label your units. Otherwise, it's not going to get any credit. And the big thing I think that I hear students making mistakes on is the type of graph as well. A line graph is going to be used a lot in science. Anytime you'd want to know, especially like these values between 6.2 and 6.7, like if I were to ask you to estimate the total expenditure in constant blah blah whatever in 1985, then you could use this graph to make that estimation. Whereas if it was a bar graph, it would be pretty useless to do that, right? And so a line graph is going to be used anytime there's like a progression, anytime there's growth, anytime there's a constant moving thing. Whereas a scatter plot is going to be used anytime the variables are seemingly unrelated to one another. Like this one is sell price and ground living area. Who even knows? But yeah. Histogram, I haven't seen. Histogram has to do with frequency, but like the frequency of a particular value. And then bar graph you're going to see a lot too, which is going to be like just comparison. You know, like we did a practice problem recently that had like different phenotypes or something like that, and it had you count them, right? And you're going to do these error bars on everything. They're going to have you do them, and so it's going to be plus or minus a particular value, and there's probably going to be a question associated with the graph that's going to have you answer, or is there a significant difference between printers and scanners? No, because the error bars overlap. And if they didn't overlap, then there would be, right? Box and whisker plot is one that you might see, but you're not going to have to draw. And it has to do with quartiles, outliers. Infinite Campus used to use these when it would post grades, and it would show you where the median quartile is, and then kind of the outliers are. Dual y-axis, or the dual y-graph, shows you two different dependent variables based on a single independent variable. Definitely have seen these, and sometimes you'll see these associated with like the three-dimensional graph. If you remember, I gave you a practice problem. It's been a while now, where you had to, it was like it was three-dimensional. And it was, I forget what it was about, like pollen and flowers or something. Yeah, well, there was a scatter plot on one year too. So, and the scatter plot was, I forget, I just gave it to my students, as a practice. And the scatter plot was two seemingly unrelated things. And then it asked you the question associated with the graph was, what is the relationship between the two? And it was a real simple answer. It was a direct relationship because it went up and to the right. It was a positive relationship. And so that you might see a scatter plot where you have two seemingly unrelated values. And on that particular practice problem, it actually gave you the variables. So it told you what to put on the y-axis and told you what to put on the x-axis. Otherwise, my guess is that you're going to see a bar graph or a line graph. That's my guess. I've not seen any of these other ones. You're welcome. Absolutely. So standard deviation. You do not have to calculate standard deviation, though it includes the. If you just started studying, you're not cooked. You know what? I actually have a ton of videos on this channel that you can watch, and they're pretty quick, and I think they're pretty good. That you can watch. I think you'll be fine, especially if you've been studying all year. Now, if you haven't been studying all year, you might want to settle for a 3, just a nice 3 that is going to be good. So yeah, standard deviation. You don't have to make the calculation. I don't know why they put the thing on there, but oh, did I get 1,000? Sweet, thanks for that. Appreciate that. Yeah, I was right at the cusp, right before I logged on. So appreciate the thanks there. Appreciate that. So yeah, standard deviation. You don't have to calculate, but you do have to think about it, right? I've not seen any questions in the FRQ or the multiple choice that ask you to tell me about this. The only thing you're going to have to do is to do this sort of thing, right? To talk about standard error. And standard error is just a way of, if you were to get a similar group of this sort of thing, this sort of data someplace else, what it's telling you is that printers would be anywhere from here to there, scanners would be anywhere from here to there. And so in another world, it's possible that printers and scanners could be equal. And so when the question asks you, is there a significant difference between printers and scanners? No, because it could be that they would be equal. Is there a significant difference between printers and laptops? Yes, because they're never going to be equal, right? And so that's the idea behind the error bars. I've never seen anything else with standard deviation or standard error other than you having to answer that kind of question. I do think and this is what this slide's talking about here. Let's talk a little bit about experimental procedures and some of this stuff. I know there's some questions on control variables and experimental variables. And I see a lot of this on a Facebook group that I'm a part of too. Some questions about what type of control variables and that sort of thing. So we can talk about that. So the control group is always going to be the one that doesn't have the treatment, right? You can think of it that way. Or we're going to give another example of a control in a moment. The experimental group is the one that's being changed, right? It's going to receive the treatment. And so the independent variable has to do with what you are manipulating. Sometimes you think about the independent variable as the manipulated variable, the thing that the scientist or whoever is changing in the experiment. What is the experiment based on? You come back over here. Let's find like this one. The dependent variable is weight. So these are weight loss trials, right? Standard deviation is important, and when you're treating patients, I'm sure, Mrs. Henson, I'm sure that is the case. Yeah, and I could see myriad reasons why it would be important, in a real scientific application like what you're talking about. But for this exam, I just haven't seen it enough to merit, like teaching it to students or whatever at length. We have an AP stats teacher that does a great job of teaching that. It's kind of funny when we talk to one another because she teaches the stats at such a higher level than what I do. And when she teaches chi-square, for instance, the students will tell her, oh, we already know what chi-square is. And then, of course, she teaches it at like 3 times higher level than what I do. I don't even know how she talks about it. So, yeah, totally, 100%. Definitely a very important. So, yeah, in this particular experiment, weight is being measured. And what is it being measured off of? Based on days, right? And so based on days are the independent variable. What's being measured is a dependent variable. There are FRQs where it'll just straight up ask you, what is the dependent variable in this experiment? And it's a simple answer. If it asks you that, so I just had this discussion with my own students. If it asks you that, don't tell me why you think that's the dependent variable. You waste a lot of time writing when you're like constantly trying to explain yourself. AP will always ask you to justify your answer every time. It'll bold it. It'll say justify this response, right? And so, every single time. And so don't justify yourself unless you're asked to do so. So if it says, what is the dependent variable, say the number of seeds that sprout, period, move on to the next part. Don't waste your time explaining yourself. That part's not even going to get read. Nobody's going to read that. If I'm your teacher, I'm not reading that. I'm reading the answer and I'm moving on, right? So don't put more than you need to. All right, so let's look at the two kinds of controls. There are positive controls and negative controls. Positive control is when you can see what I wrote down here for the definition. Exposed to a treatment with a known result, right? Not exposed to experimental treatment. And so I'll use Griffith's experiment. Griffiths did experiment on mice and pneumonia back in the 20s and 30s in order to, we eventually discovered genetic transformation, which we've talked about. And he injected some of the mice with a bacteria that he knew caused pneumonia, right? Why did he do that? Well, it was a positive control, right? He was establishing norms for the experiment. And so a positive control is still a control, right? Because it's still being used as a basis of comparison, but it's something that you know is going to happen, right? It's in order to compare unknown things, right? So in this particular experiment, the unknown was what's going to happen when you mix the rough strain, heat-killed, smooth strain together, right? That's the unknown. All these other things were known treatments, and that's the idea, right? So positive control is when you have a known treatment or known variable. A negative control is, what does virulent mean? It's a good question. Virulent means like it's able to cause disease, right? That it's able to spread disease. If Mrs. Henson is still in the chat, she could probably give a more technical definition of that. But that's probably close enough for us. It's able to cause disease. And something can be more or less virulent as well. It can cause disease more rapidly or less rapidly. So think of it as like viral, like a viral video or something like that. It's a way of thinking about it. So negative control is not exposed to any treatment. And I feel like I need to replace this picture. I feel like I remember it all the way back in August that I thought this picture was bad for this. And so here we are again. Should have replaced it then. How much ecology should you know? Well, you should definitely know all the ecology that is in the exam description or course and exam description. But the good thing about ecology is I feel like, and my students kind of echoed this sentiment as we talked about it, that as I was teaching through the ecology unit, it really felt a lot like I was teaching the evolution unit again. So, because it has a lot to do with behavior, it has a lot to do with response, and so yeah, that's a way of thinking about it. It's not, honestly, it's not that bad. My ecology exams were some of the highest. Still hasn't finished teaching it, so I just want to know. Actually, I just made a video that I review the whole unit, and I do it in like 26 minutes or something. Unit 8, ecology review. So if you want to check that out, go through it pretty thoroughly. And it's not a hard unit, honestly. There's a couple of calculations that could come from it, like population and that sort of thing. But it's honestly just not hard. Like, it's just, can you like read a problem and fill in the right numbers where they go? I think the hardest part about those population equations is knowing that the dn over dt is a number. It's not, it's its own thing. I don't know how to describe that. So yeah, negative control. I'm gonna need a new picture for that one. Let's go back up here. The one that's not getting water, it's a negative control. And then control variables, these are things that you keep parts of the experiment intentionally left unchanged. Yeah, there's that. Do I need to go over chi-square? Maybe you can tell me in the chat. Do I need to go over chi-square? Can I just start in unit 1? And because I'm happy to go over chi-square and talk about it. But yes, please. Okay. All caps. No punctuation from Mr. Urali. We will go over that. Why chi-square? Well, so what is the purpose of chi-square? Purpose of chi-square, yeah, go over degrees of freedom. Yes, absolutely. Can we go over negative, after this, can we go over negative control again? I will go over negative control again. Right. All right, so, and maybe I might be able to work negative control actually into my discussion on chi-square. Let's see if I can pull that off. No, I probably won't be able to, now that I think about it. Okay. So what is the purpose of chi-square? So with chi-square, you're going to have an expected set of values, right? So M&Ms are a perfect example. So what you see here, this is M&M's color distribution according to their website. So according to the website, M&M has a particular color distribution, and there it is. So meaning if you were to get 1,000 bags of M&Ms and measure them, then you should get these numbers right. You should get them. How do you know that the numbers that you actually get are close enough to these numbers so that you can just accept them, right? That you can say, yeah, it's close enough. Or how do you know that you don't have somebody working for you that really hates yellow M&Ms and they're purposefully trying to remove them from the factory line so that there's more red than there should be? Right, and so that is the purpose of chi-square, is to determine whether or not a set of values are close enough to what you expect, right? And so that's the whole idea behind it. Are they close enough to where any difference is just random, or is there some significant difference that you would require more questioning, right? So why are they that different? That's the idea behind it. Typically not going to be M&Ms. Typically, when you see chi-square, you're going to see it associated with heredity, like Hardy-Weinberg or like Punnett squares or something like that. Though I've seen it in all kinds of applications. You're definitely not going to see it with M&Ms. All right, so here is the so it's goodness of fit test is another way of thinking about, another quick way of saying what I just said. So here's the chi-square equation. This little c right here is not going to be on your formula sheet. I don't know why it's there, so just ignore it. So a couple things here that you need to know. First of all, chi-square is what you're solving for. It is not, you do not need to take the square root of that. Ever. Resist. I know when you see that little 2, your math training has taught you to push that little button on your calculator, and now you're done. Now you have it. But that's not an x. That's a chi, right? And chi squared is what you're going after. And so when you get that number, be done. All right? And so here's sigma. Sigma means sum, right? And so what are you summing? Well, you're going to do this a bunch of times. You're going to do this center thing. Can I draw here? I think I can. Yeah, you're going to do that a bunch, right? And how can I erase? Eraser. Okay. So you're going to do this a bunch. What is this? Well, let's go up here. So these are my expected values, or E. Well, let's say that my observed values are, I don't know. I'm not going to do the whole problem here. Yeah, you can think of it that way. As just resist the urge to do that. Correct. And so, you know, if you had 12, let me find my drawing here, not drawing, but put letters in thing. I'm used to the one at school. I don't know where it is, so I'm just going to be stuck drawing things. Okay, so this is going to be bad. So let's say that brown is 12 and yellow is 13, It's bad. I'm not going to do this. I'm going to like just talk about it, right? Instead of actually doing any math, because I'm just not set up. Maybe tomorrow I'll be a little set up. I think I'm planning on doing this again tomorrow night before the exam. All right. So my expected is 13, but my observed is 12, right? And so observed minus expected is 12 minus 13, which is -1 squared, which is 1 over expected. So 1 over 13 is a number, right? And so that is one thing. And then I'm going to do the same thing for yellow, the same thing for red, the same thing for blue, the same thing for orange, and the same thing for green. And so let's say, I don't know, let's say I add all of that up and my number is 7.27, all right? And that's my number. And that number is my chi-squared. All right, so what do I do with this number? Well, I'm going to compare it to my critical value, right? So what is that? Well, usually you're going to be asked to use the p-value of 0.05. We're not going to talk about p-value, not because it's not worthy to be talked about, but because, it's not talking about it. It'll be given. It's not a stats class. Again, this is AP Bio, so we're not going to talk about what that means. And then degrees of freedom. So what are degrees of freedom? So the way that I demonstrate this in my class is, so you have 6 things. And so imagine that I had a hand of like 6 cards, right? And I'm holding it out. And there's 6 people there. And I'm going to take my hand of cards and I'm going to hand it to the first person. They get a choice, right? There's 5 cards left. I give it to the second person. They get a choice. And I'm going to keep going down. The second to last person, there's 2 cards left. I'm going to hand them. They get a choice. Does the last person get a choice? They don't. There's 1 card left. They don't get a choice. And so in reality, even though I had 6 cards and 6 people, there were only 5 choices that were made, right? And so the degrees of freedom were 5 in this case. How do you do that without thinking about people taking cards? Well, it's just 1 minus the number, or subtract 1 from the number of things, right? So there's 6 colors in this case. Take 1 from that, 5 degrees of freedom. And so I have my 5 degrees of freedom here, right? 5. I'm going to use 0.05 here. The highlighter would be so much better. Here it is. And so 11.5 or 11.07, I don't know where I got 0.5. 11.07 is my critical value for the problem that I just came up with. We call this the critical value, right? And now what do I do with that critical value? Well, I compare it to my chi-square. My chi-square is lower than my critical value. And so what do I do? Well, if my chi-square is lower than my critical value, I accept the null. Now, there's a lot of people that will really hate that terminology, and they'll say, no, what you should say is you fail to reject the null. Who cares? It's not that important. And so what do we mean when we say we accept the null? So our null hypothesis is that any change from the expected value is random, right? That means that the numbers that we got were close enough to the expected that any difference is going to be random. If my critical value had been higher than 11.07, then I would have to reject the null hypothesis, meaning that there's some other alternative to the reason why those numbers are higher. Maybe there's somebody that works in my factory that really hates yellow M&Ms and just whatever, who knows, right? And so that's what you're doing there. And so you may be asked to give a null hypothesis for an experiment. And so let's go back to this one up here, this one, right? So the null in this one would be that, think those same lines, right? That the change that I'm making isn't going to do anything. So like the null here would be that the water isn't going to cause it to grow more significantly than the control group. Yeah, right. They didn't. So we failed to reject the null. They didn't want to get rid of the yellow M&Ms. It was just chance. That's right. Right. And so the null hypothesis here is that the water wouldn't cause it to grow any more than the control group. But if the experimental group is significantly higher than the control group, then we have to come up with an alternative hypothesis, which would be something's causing it to grow. We know it's causing it to grow. The water's causing it to grow, right? And so that's the whole idea behind that. And so here's an example that I gave, and I did this one, and it looks like I was trying to, this is probably my actual handwriting, not me writing with a mouse, which doesn't look a whole lot different. My handwriting's disgusting, but that's okay. And so in this particular cross, right, we have expected results, and those results would be 25% big R, 25% little r, 50% big R, little r, right? And so those are our expected, which is what I wrote here, and these are our actual or observed results. And so we do chi-square, and this was our chi-square 28.38. Since there's three things here, how many degrees of freedom do we have? We have 2 degrees of freedom, right? And so we'd come up here, 2 degrees of freedom, critical value would be 5.99. 28.38 is much higher than 5.99, and so we would have to reject the null hypothesis that there's something else causing this besides our normal pattern of heredity, right? And so that's the way that you would think about that. Okay, real quick, I'll go back to negative control, as was asked. Negative control is just this when a treatment is not added to. So in positive control, there's a known value. Here is the example of the negative control where water is not added, and so it is a basis of comparison. The water is not added to the experiment, showing that it's a control group, but it's negative, meaning it's not given that particular treatment. Positive control is given a treatment, but it's given a treatment with a known result. That's the big difference. All right, I'm moving on past this Unit 0 stuff. I'm going to keep going unless there are any particular question. All right, so good question. Normal control. So when you think of controls, there's negative and positive control. A negative control is what we typically think of as a control group, right? Like you've probably been taught control group your whole life in science to some level, and that control that you've normally been taught is a negative control. The positive control is the new idea, really, for AP Bio students, and then adding negative to the what we would call the normal control in this case is what causes it to be a little confusing. So in this case, adding water at all, in this particular experiment, makes it experimental. Now, what if you knew that this particular plant grew, that 9 out of 10 seeds would sprout every time 200 milliliters of water is added to it? And so you added 200 milliliters of water to one group, and you added 700 milliliters of water to another group. Then the 200 meters would be a positive control because it's a treatment with a known outcome. The 700 would be your experimental group because you don't know the results of that, and you are trying to discover the results of that. Right. Questions? All right, I'm moving on. 1.1. We're just getting into Unit 1. Absolutely. Glad to do this. I'm really thankful that the chat is really calm and there's not a bunch of people like saying Skibbity, Ohio a bunch in the chat. So thank y'all for not doing that. Because it was pretty rough. The guy who teaches right next door to me he's like some sort of famous chemistry teacher, I'm assuming, and his live chat was kind of funny. So, and then the one I watched on the history guy, that was really funny too. So yeah. Okay, Unit 1, structure of water and hydrogen bonding. I talk about emergent properties. That's kind of an activity that we do the first day of school. Cost of Fame, Israel. I saw the poor history guy. Yeah, he was pretty rough. It was pretty rough. I mean, he handled it really well, though. He handled it really well. And I stream quite a bit because I play video games as well, and I stream. So I'm used to people writing weird stuff. So it doesn't bother me. And I guess those Well, I know the history guy Heimler is probably used to that too, but I think my next door neighbor, Mr. Krug, wasn't quite prepared for that. So that's right. Next time he does it, he'll be ready. He'll be ready. All right. So let's talk a little bit about structure of water and hydrogen bonding. Hopefully you know a little bit about this stuff. Hydrogen, not hydrogen. What am I saying? Protons, electrons, neutrons, all that business, charges. We're gonna spend any time there. All right, structure of water, pretty important. All right, so the water molecule, obviously a very important molecule, and the way that water behaves is very important for the body. And so water's obviously one oxygen, two hydrogen atoms, and they're covalently bonded. What does that mean? So that means that they share electrons, and you can actually see that really well here. Oh, what happened? Okay, here we go. Zoom in a little bit. So you can see that really well here, right? You have this nucleus here that has 8 protons in it. You have these 2 nuclei here that have just 1 proton each. And together, there are 10 protons in this water molecule, and they are sharing these 10 electrons, right? Hydrogen brings 2 to the table, 1 each, and oxygen brings 8 to the table, 10. And because they're covalently bonded, they are sharing. Co means share, valent, outermost electrons. And so they're sharing their outermost electrons. Well, notice how much bigger oxygen is than hydrogen, right? The oxygen atom is 8, or oxygen nucleus is 8 times more positive than the hydrogen nucleus, right? And what do electrons like more than anything? They like protons, right? And so these electrons are going to spend the majority of their time around this oxygen nucleus as opposed to this hydrogen nucleus. And so what that's going to cause is it's going to cause the unequal sharing of electrons. Do you have to memorize all the organelles and plants? No. There's actually not a lot of memorization on this test. Typically, when you're going to, let's pretend that you're given a question about organelles and plants. In my experience, you're not going to get a question like, what is the organelle that does this? Those are the questions that we all want, right? Because those are like the 7th grade type, like memorized questions. But what you're going to get instead is like a paragraph that's telling you about some obscure new plant organelle that actually had a student tell me about this morning, called the nitroplast or something like that, and it's going to ask you all these questions about it, right? That you're going to, so it's less memory and more application of things that you're being told. Knowing the information is good because that's the first step on the way to applying that information, but memorizing it is not all that helpful, at least in my experience. So, but there's stuff out there to help you do that. Yeah, you mean knowing it's going to be good, but applying is much better. And so because the size of these molecules are so different, right, there's an unequal sharing. It's always, the story that I always tell is like if I'm on a trip with my family and I stop and I get a bag of gummy worms or gummy bears and pass them to the back, there's no equal sharing of the gummy bears. There's like negotiations going on back there from the moment the door is shut and the moment the gummy bear bag goes to the back. The intent isn't that they're not going to be equally shared. There's a power struggle, right? And so there's a power struggle with these electrons, and the oxygen, because they're bigger, is going to get most of the electrons, and because of that, it's going to have this slight negative charge, right? So if it's slightly negative, what is the hydrogen going to be? Well, it's going to be slightly positive because the electrons aren't there anymore. And so it's still shared. The electrons are still being shared, but it's an unequal sharing of electrons, and we call this unequal sharing of electrons polarity, right? And so when we talk about the water molecule being polar, we are saying that this is why, right? It's not that there's like free electrons floating around. When we talk about water having a charge to it, we're not talking about like electrons like sticking off, forming bonds with things. They are going to form bonds, but not in the same way that this hydrogen and this oxygen are forming a bond with one another. They're not both hanging on to the electrons, right? They're just kind of attracted to one another, which we're going to talk about in just a moment. Not yet. So this shows you some polar, but this shows you polar molecules that have this kind of positive negative thing going. Non-polar is where there's an equal distribution of charges, or you could say an equal sharing of electrons. I'll let the Mr. Krugs of the world teach why and how and numbers associated with all this beyond the scope of my class and frankly beyond the scope of my knowledge. And so I'll let them teach that business. Speaking of that, if you're taking AP Chem, you should check out Mr. Krug's YouTube channel. He's pretty popular. You probably already checked him out. All right. And so hydrogen bonds. What is a hydrogen bond? A hydrogen bond is a bond between two polar molecules, right? So here we have one water molecule here, right? And here we have another water molecule right here. And the oxygen, remember the oxygen is slightly negative, the hydrogen's slightly positive. That positive on one oxygen or one water molecule is attracted to the positive on the other water molecule, the negative here, sorry, the positive here, they're attracted to one another. And this attraction is called a hydrogen bond. There's no physical connection as far as electrons. They're not being transferred. They're not being shared. There's just an attraction. Think of it as like a magnet almost. You know, when I teach this in class, I'll pull the eraser off of my whiteboard and then I'll put it back on. The whiteboard has an obvious attraction to that eraser, but it's not the same as the screws that are holding the whiteboard to the wall, right? That's a mechanical connection that is strong. Whereas this is a weaker one. Are the bonds between the oxygen and the hydrogen on the same molecule, covalent bonds? Yes. So, right, good question. So this is a covalent bond, right? This is a covalent bond. It's a covalent bond because this hydrogen here and this oxygen here are sharing electrons. This bond right here with the red arrow is a hydrogen bond because it's two separate water molecules and they are attracted to one another through a hydrogen bond. So this molecule's keeping all of its electrons, this one's keeping all of its electrons. There's no exchange of electrons between the two. They're just merely attracted to one another. All right. And this molecule is pretty much why our whole body works. Or this molecule, this bond is why our body works the way that it does. Hydrogen bonds, the most important bonds in living things by far, not close. Obviously, covalent bonds are important, but the reason that life works is because hydrogen bonds are the way that they are. And here's some other examples. Okay, so this one shows you how one water molecule can form bonds with 4 other water molecules. Do you have a choice on what graph that you make on the FRQ? You do. You do have a choice on the graph that you make. However, only one of those choices will be correct. And so you need to make the graph based on what you know. So like if the independent variable is something like temperature or time, usually it's going to be a line graph. But if it's, I wish I had some examples. Maybe tomorrow I'll post some examples when I stream tomorrow. I think that would be helpful. I can't use, like continuous, thank you, good. I can't use like the provided materials. I can't show them. But categories, graded way of thinking about it. Categories, comparison is the way I think about it too. Like, like the picture that I had earlier, comparing printers to laptops to copiers. Those are the main two. You might see a scatter plot, maybe. I'm doubting it though. You're probably going to see a line or a bar, more than likely. I believe last year's was, I don't remember last year. I think it was a bar graph. I don't remember. So anyway, good question. Make sure, access titles to get the points. Absolutely. Big deal. Scaling. The graph is actually worth 3 points. I guess I could have said that. So you get a point for correctly titling and your units and everything. You get a point for scaling, and then you get a point for the correct placement of your plots, correct plotting of your graph. I think it's the only word. It's like the exam uses all these bolded terms, right? Hopefully your teachers have shown you FRQs, and you've seen these bolded terms a lot. Every bolded term is a point except for the word construct, and it's always 3 points. At least in my experience. I don't think they're going to change it any, but you never know. They like to mix things up. All right, so this oxygen, or this water right here, can form hydrogen bonds with 4 other water molecules. All right, so that makes water act kind of weird. Sorry if this sounds like a tone. Oh, that's a good question. I'm glad you asked. So the FRQ section is actually separate from the multiple choice. So I think it's part of the same booklet. Like, I don't think you get a separate booklet. I've never taken one either. I've seen a bunch of practice ones, but the only practice ones I've seen are like PDFs. And so it may be a separate booklet, it may not be, but it's all together. So you'll like see the prompt, and then there'll be some lines underneath that for you to write your response. And I will say this about the lines. Just because it gives you 2.5 pages of lines, it's not expecting you to use all those pages. In fact, don't. Like, there's not anybody that exists that could fill out all the pages in the time allotted. No one can write fast enough. So it's, I tell my students, I don't know where people who are watching me are from, but my students have to do these tests in middle school where they're literally told to fill the whole box in with words. And I always tell them, don't resist, don't fill the box in, just answer the question. So yeah, you will be given plenty of space to write underneath the prompts. Separate booklet. Yeah, in the past I've been, like, we used to order the old, like, the exams. Yeah, absolutely. Glad to help. Yeah, and tomorrow night, I may actually talk a little bit about the exam. Just my students, I've talked about it all year, but I will do that. I take for granted that everyone's heard about this test that you're getting ready to take. So yeah, I hate that if you haven't. But yeah, some people, you may be taking this independently or something too. So yeah. So let's talk a little bit about what water's polarity causes it to do, right? So what does water's polarity cause it to do? Well, it causes it to be really attracted to itself. Because it's really attracted to itself, it has the property of cohesion. All right, so this cohesion is the idea that water is really attracted to itself. And you see this in particular when it's up against something that it's not attracted to. So like the surface of a leaf is going to have a nonpolar substance on it, like a waxy cuticle. And the water is more attracted to itself than it is to the wax, like infinitely more so. And so it will bead up like this. But there are things that the water is attracted to, right? Aside from itself, like glass. And so what do we call this? Well, we call it adhesion. Remember back when we were all wearing masks, and if you wore glasses like me, this is what you had to deal with, because the steam from your breath would like fog up your glasses because the water in your breath really liked the glass, right? Because it's adhesion. And you mix these two things together. So you mix the fact that water likes some things and water really likes itself, and you have this thing called surface tension. I was getting at something else. There it is. Capillary action. All right, so capillary action is water's ability to pull itself up tiny tubes. How can it do that? Well, it likes the tube, right? So it pulls itself up the tube, and it likes itself, so it is pulling itself up too. Why does this matter? Well, if you've ever been to the nurse and they like take your blood, they'll put this little glass tube up and they'll like get all the blood up in that tube, right? And that's how they do that. Is because the blood wants to get in the glass, because blood is mostly water. And trees use this to get water all the way to the top. Capillary action, adhesion, cohesion. Let's go back up here. talk about surface tension. Surface tension is the phenomena that is associated with the fact that the way that water is attracted more to itself near the surface. Why is that? Because near the surface, water would want to like come apart, right? And so it doesn't. And so it's more tightly connected to itself there than it is anywhere else. And some animals use this to their advantage, like they skate on top of that water. Surface tension, these are supposed to be moving. That's why they look weird. They probably are moving on my actual PowerPoint, but this is OneNote, so they're not moving. That's why we use soap, by the way, because soap actually decreases the surface tension of water and allows water to work. So like when you use detergent for your clothes, water can actually get through the fibers of your clothes because soap causes the surface tension to break. There's your factoid for the day. Solvency, water's ability to break down things. How can it do that? Well, it has a charge. And if those other things have a charge, it tears them apart, right? So water, this is salt. Salt is made of positives and negatives. Water likes positive things and negative things, and so it tears them apart. It breaks up the ionic bonds between sodium and chloride and separates them. Not like permanently. It's not like you're going to have chlorine gas coming out of solution or anything crazy. I don't really understand it 100%. But I do know if you take all the water out, what do you have left? You still have the salt, right? So it just kind of breaks the salt down. And then water is more or less dense as a solid. How is that possible? Well, the reason it's less dense as a solid is because as the water cools, the molecules will slow down. And as they slow down, they kind of like lock into place. It's kind of the way I think about it. It's like having a bunch of like little beads or something in a tray that has a little spot for them, and you can like jiggle them and stuff, but when you slow it down, all the little beads will find their spot and like set there. And when they do that, they kind of like lock into place, right? And they spread out when they do that, and that causes so they take up more space, but they're still the same amount of matter, right? And so then that is if you increase the denominator, which is volume, but keep the mass the same, then you're going to decrease the density, which is exactly what happens here with ice, which is good for as far as living things are concerned, because ice floats, all the ice will go up to the top of the pond or whatever they're living in, the ocean in this particular case, freeze the top, and the bottom will stay liquid so that the fish and things can live. If ice sank, then the whole pond would freeze and kill everything, which wouldn't be good. And because water is able to bond with four other molecules, it has very high heat capacity, meaning that it takes a lot of energy to break this system up, and so it can hold a lot of energy. You know what? I think that's all of 1.1. It took me a while to get through that, but that's okay. Any questions about any other questions too? I can pause just if there's just particular things that you want me to talk about, because you guys have kind of come in with random questions, which I'm 100% fine with. But I've just kind of been plodding along because I didn't know what else to talk about. But if you have any other random questions, please ask, or if you just want me to talk about a particular section of material. Difference between cohesion and adhesion. Good. So cohesion is specifically when you're talking about water's attractiveness to itself, right? Versus adhesion is when you're talking about water's attractiveness to other things. So here's a great way of thinking about it. Think of adhesive tape. What does adhesive tape stick to? Most things, right? Have you ever heard of cohesive tape before? There's a thing called cohesive tape. You know what cohesive tape sticks to? Only itself, right? It won't stick to other things. It only sticks to itself. That's like, like if you play sports, you can like wrap your pre-wrap that you like wrap your ankle with or whatever. That's cohesive. It's a good example. Yeah, good question. Are covalent bonds stronger than hydrogen bonds? Yes, significantly. Significantly stronger than hydrogen bonds. What property of water causes surface tension? Polarity, 100%. I may have told you wrong about that. But yeah, I think it's been different in the past, so I don't know. It may be different this year. I know in the past the students actually had a separate FRQ book, and they would actually send us those books. When is everyone taking the test? I think everyone's taking the test on May 16th in the afternoon. Unless you're not from here, or unless you are having to take a different test. Yeah, Thursday at 12. That's when everyone's taking it. Or sometimes people will take a makeup test, which I don't know when the makeup test date is. But yeah. It's a good question. What other questions do we have? I don't remember what 1.2 was. The elements of light. Wow. I always choose the best pictures. This is straight up just, that thing is smiling. It's just straight up photoshopped. And I have no idea why I chose this picture for this particular concept. How much time should I spend on a multiple choice? Good question. So there are 90 minutes for 60 questions. And so just raw averages is 90 seconds per question, right? 1.5 minutes per question. However, some questions are going to take you 15 seconds to answer. And so then all of a sudden, the next question, you have 2 minutes and 45 seconds to answer that question, right? And so you need to think about it that way, because some of the times you're going to get a question that takes you 15 seconds, and some of the times you're going to get one that takes you 3 minutes. And so when you get to the 3-minute question, you don't need to panic, because you need to think about it as you've kind of banked time, right? You're constantly banking time when you take a test. You don't need to sit there and actively think about how much time you've banked, because you need to be thinking about answering your questions correctly. But hopefully there'll be a time or a clock or something in the room that you can kind of pace yourself. It's like if you start at a certain time, just know, okay, at this time on the clock, I should be this far into the exam. 45 minutes in, you should have 30 of the questions answered, right? And if you're ahead of that, then you can kind of slow down or not. If you're behind that, you may need to pick up the pace a little bit, because you don't want to get to a place where you have blanks left. You want to try to answer all of them, Yeah, do I guess you just absolutely. You're not penalized for wrong answers. You should. Yeah, you should definitely do that. Pick a letter. Pick one of the letters and just do that one every time. Don't random. Just pick A or whatever. How should you manage your time? Oh, I'm glad you asked that question. I'm actually going to just do this. Let's see. Oh, there's a 2023 FRQ. Let's see if I can. This is actually public release. So here, this is perfect. This is perfect. This is exactly. So yeah, 2023 FRQ. This is public release information. And so this is what you're going to see at the beginning of your FRQ. Questions 1 and 2 are long free responses, require 25 minutes each to answer. So again, that's just a suggestion, right? It's not saying that you ought to spend 25 minutes on them. It's saying that you could, right? I think that you can do them quicker. In fact, we did question 1 in class the other day, and most students were done with inside 20 minutes. And so I think you can answer that one in 20 minutes. And then the questions 3 through 6 are supposed to be 10 minutes each. And so let's look at these together. And I actually made videos of me answering each one of these. The video of me answering this one has actually been fairly popular recently. I don't know why. Probably because it's question number 1. I'm not going to go over these specifically, but just kind of show you the exam. And so here's a great example. And typically questions 1 and 2 are around 10 points each, and questions 3, 4, 5, and 6 are 4 points each. And I said around because this first question is going to give you an example here. So you can literally count the points here, right? Let's count them. Here's 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is a 9-point question. That's it. right? They're not going to ask you more than this. And so like earlier I was talking about a question that, look, identify a dependent variable in the researcher's experiment. Literally just say what it is and move on. Don't tell them what you think about dependent variables. They don't care. They just want that. They're looking for that, and then they're going to move. They're not even going to read your whole sentence. Promises. Other teachers grading this, and teachers don't read every word unless they want to just not grade tests on time. It might take like a month and a half to grade all your tests if you're reading every word. So here's 1, 2, 3, 4, 5, 6, 7, 8, 9 points, right? Calculate. Some of these are really simple, like identify, calculate. Very simple kinds of answers versus justify. So this is when they want you to justify your answers, when they ask you to. Before, other than that, don't. So let's look at this one. So here's an example. Someone was asking about like how do we know what type of graph to use? This is a perfect example. And so I don't even read. You can read the prompts. Actually, you should read the prompts because you're better readers than me. But when I see this, I know that I'm going to be asked to graph this. And sure enough, it doesn't give me the graph on here. But species 1, 2, 3, 4, 5, 6, and their different mitochondrial levels. This is a bar graph because I'm comparing species 1 through 6. 100%. That's what's going on, right? And this is a interesting bar graph because each species is going to get 2 bars, right? Species 1 is going to get a bar for normal CO2, and species 1 is going to get a bar for elevated CO2. And so I would set up my x-axis to have 6 groups, and each group would have 2 bars. That's what I would do. And so let's see how many points here on this one. 1, 2, 3, 4, right? Because construct is 3. 2, 3, 4, 5, 6, 7, 8, 9. So those are 2 9-point questions right there. Do we have to do this in pen? Great question. Yes, you do. You do have to do it in pen. Why do you have to do it in pen? So this is another good question. Because the tests are scanned. And I haven't even talked to my own students about this, and I need to do this. The tests are scanned. And so when you take your test, they're not grading your original. They're seeing a scan of your test, and pencil doesn't scan really well, right? And so pencil doesn't scan really well, so pen does. However, on this graph, I would do your graph in pencil and then go back. Once you're happy with your graph, then go back over it in pen. That's what I would do. Can you explain what the difference between independent, dependent, and control is? Yes, I can. Dependent variable, what's being measured. And so in this particular experiment right here, what's being measured? ATPase activity or relative amounts of mRNA. How do I know? Well, I could read the prompt, and the prompt would help me with that, right? And the independent variable is what are those measurements based off of? Well, in this particular case, they're based off of the type of mutation. Is it wild type? Is it this or is it that, right? And so this would be the independent variable. Here, independent variable would be the species, right? It's based off species 1, 2, 3, 4, 5, 6, and what's being measured, the number of mitochondria in normal CO2 versus elevated CO2. And so my x-axis is always going to be my independent variable, and my y-axis is always going to be my dependent variable. Every single time, ask yourself what's being measured. That's going to go on your y-axis. What is that measurement based off of? That's going to go on your x-axis. Phil and Chippy, I always do. So yeah, good questions, really good questions. So yeah, you have to do it in pen because your test is going to be scanned, and you want the grader to be able to read your responses. If you do it in pencil, are they going to grade it? They're going to try. It's not like they're going to throw it out, but it's just harder because it's scanned, right? So let's look at this question. This is a 10-minute question, right? You can read this prompt about sand lances. In fact, you should. But let's look at these questions. Let's just think about this, right? This is a good one because this is supposed to take you 10 minutes. Let's just do it without reading the prompt. See if we can pull it off together. Describe the effect of increased biodiversity on the resilience of an ecosystem in a changing environment. Literally don't need to know anything about sand lances in order to answer this. Increased biodiversity is going to increase the resilience of an ecosystem, period. That's it. Why? I'm not asked to justify my answer. It's going to increase the resilience of that. Now, if I was asked to tell why that is, then I might go into more, but I'm just describing the effect. It's going to increase. Justify your response, then I won't, but it's not asking me to do that. So I did part A without even knowing anything about sand lances. Justify the scientists selecting 5 degrees Celsius as the lowest at 400 micro ATMs is the lowest. And so then you may need to go up there and read the prompt, right? I'm not going to answer this question because I haven't looked at it at all. State the null hypothesis for the experiment. Again, you do need to read the prompt for this, but it's not content based, right? It's not anything about sand lances or anything. You should be able to do this just by reading this experiment. And again, I have like videos of me going through all of these questions, so that may be helpful to you. Scientists claim that a reduction in the population size of Amodides sand lances will affect the stability of the entire ecosystem. Please provide reasoning to support their claim. And so this is kind of linking to D, because here you said it would increase the resilience of the ecosystem, and here they're saying that if you take them down, it will affect that. Why is that the case? Well, you could talk about it being an important part of the ecosystem, and if you remove a part of the ecosystem, the whole food web will kind of fall apart. Talk about how all of them are grouped together. It's the talk about the null. Talk about part C for what is the null hypothesis? You mean to describe the control group maybe from up here? So in this particular one, it'd be normal. The normal CO2 would be the control group. I'm sorry, I'm not following your questioning. Ooh, this is a mess right here. Non-cyclic. So yeah, here's an example of something. I'll talk about this one too. It's a 9:05. I'm starting to feel the weight of the day on me. But I have enjoyed this quite a bit. I'm definitely planning on streaming tomorrow night, and I'll probably try to stream a little longer tomorrow night as well. But here's an example of something that like kind of freaks people out when they see it. Non-cyclic electron flow for B. Sorry. Yeah, that's probably correct for that. Honestly, I haven't really thought about it, but yeah, it probably is. Serve as a baseline for the measuring. Non-cyclic electron flow and cyclic electron flow are two major blah blah blah, and then it talks about non-cyclic electron flow. This was one of those things that when teachers saw this question, they were like, "Oh no, we haven't been teaching non-cyclic, or we haven't been teaching cyclic electron flow, and do we need to go back and teach it?" No, because you should be able to like see this picture and kind of discern what's going on, right? And again, this is a 10-minute question. Describe the role of chlorophyll in photosystems and plant cells. You don't need any of this to do that. It's just straight content. Describe the role of chlorophyll. It's a pigment that absorbs light. Moving on. That's it. Yeah, ETC. Good. That electron transport chain. Oh. All right. Based on figure 1, explain why the increase in ADPH or increased rate will cause an increase in the flow of electrons through the cyclic pathway. Because the electrons are being reduced, because it's just being let go. I mean, I love the ETC. It's wonderful. It's just wonderful. Right? And so this one shouldn't take you long. These two are just simple content. And right here you actually have the mark on you had to mark on this. So one of the questions, 100%, is going to have you mark on the question itself. And so make sure that you pay attention to that. Let's see how it asks. I forget. Part C here, yeah. Can we go over cladograms? Yes, but we'll do cladograms tomorrow. I'm kind of running out of steam for tonight. But yeah, we definitely will. We can go over a question like this tomorrow. Yeah, I'm going live tomorrow. I'll probably go live. I may do that a little earlier. I said 8, but I may do it a little earlier, and then and go later, go into the night. Not into the night, but yeah, you guys need to go to sleep too. But let's see, using a template in the space provided for your response, represent the point. And so you're going to see this word too, and what it's asking you to actually go up here and do is then draw on this thing. And so make sure that you do that when they ask you to do that. One of the questions will for sure ask you to do that. I think this question was hard. Yeah. All right, so this has been fun. Appreciate you guys being a respectful group and asking good questions. But I will be coming on live again tomorrow. Tell your friends. Tell the people that you are taking the test with. And we will join up again tomorrow. Bring your questions, please. Definitely we'll go over cladograms. I'll probably spend some time on Unit 7 and 8 because I know some teachers have a hard time getting to those units. And I'll talk about any other problems, any other questions that you guys have. All right. It's been fun. Thanks a lot.